Hello friends, we are ready with our new topic, control of microorganisms. We all know microorganisms like bacteria, viruses, fungi and protozoa. They are widely present in the environment, they are all around you. Some of these microbes are beneficial and essential for the processes like digestion, nutrient cycle. Even in biotechnology, we find them very useful. While still there are microorganisms which cause diseases, which are responsible for food spoilage and material damage. So, controlling these microorganisms, controlling their growth and spread is a crucial point for maintaining human health, ensuring food safety, preserving materials, and preventing infections. See, when we are talking about the control of microbes, it refers to the methods used to reduce or inhibit, or even we can say eliminate, the microorganisms to a safe level. These methods they aim to either kill the microorganisms or we can prevent their growth. Means both the measures can be taken. Either we are going to kill them or we are going to prevent their growth. The growth will be inhibited. So microbial control is especially important not only in the research laboratories or in hospitals, but they are equally important in food industries, in water treatment systems, in pharmaceutical preparations. Even in your home, you may find that the control of microorganisms is being practiced at every level. So there have been various methods which we employ to control the microbes, and the choice that which method will you follow will be based on your need. So in this whole topic we are going to discuss about the various control methods of microorganisms. Now before we begin in this journey to control the microorganism, what different factors are we going to follow? First of all, the clear understanding of the terminology is very important, as when we discuss about the control of microbes, because the word as such we use like disinfectant, antiseptics, they are often confusing. We use them interchangeably, which is not correct. So the control of microbes, it refers to the methods that are used to either destroy, inhibit or remove the harmful microbes to prevent infection or contamination, uh, or we can say the spread of any kind of disease. Now there have been various terminologies which are displayed on the screen. The first one is sterilization. Second one is disinfection, sanitization, antiseptics, chemotherapy, microbicidal and microbistatic. So all these terms, they seem similar, but actually they are not. They have a little difference among them, and that is important to understand if you are a student of science. So first we will take up sterilization, which is a process by which all living cells, even they are spores or they are acellular entities that we call as virus, are either destroyed or they are removed from an object. So when sterilization is achieved using a chemical agent, that chemical we call it as a sterilant, and the sterile object will be free from any viable microorganism, spore, infectious agent. Means nothing would be present on the surface of the object which we will sterilize. So sterilization refers to complete removal of any kind of microbe. Now if we talk about the disinfection, disinfection is actually the killing, inhibition or removal of microorganisms that are not safe for us, means which can cause the disease, or we can say it is the substantial reduction of the total microbial population and the destruction of potential pathogens. Means we are not going to kill all the microorganisms, but only those will be killed which are pathogenic for humans or which can cause any kind of infections. So similar to the sterilant, disinfectants are also used, which are the chemicals which are used for the process of disinfection. So generally disinfectants, the chemicals which are used to cause disinfection or to reduce uh, the total microbial population or the destructing pathogens, they are used on non-living or inanimate object. It does not sterilize the object, so the viable spores or the few microbes may be remaining in case of disinfection. While sterilization, as we just now saw, it is the complete removal of microbes. The third the third term is the sanitization. In this, the microbial population is reduced to levels that are considered safe by public health standards. And the inanimate objects, means the non-living objects, can be cleaned as well as they can be disinfected by the sanitization process. To control the microbes on or in the living tissues, we should have some chemical agents. So till now, whatever we have discussed, the sterilization, disinfection or sanitization, they are used on the inanimate objects. But for control of microbes on or in the living tissue, we again should have some chemical agents, and they include the antiseptics, and these antiseptics, they causes antisepsis, right? And the antisepsis is the destruction or inhibition of the microbes on a living tissue, interestingly, and it is somehow different from the above three terms. So it destruct or inhibit the microorganism from a living tissue to prevent the infection, or generally we call it as sepsis. So antiseptics are also chemical agents, but they are applied to the living tissues in order to prevent the infection. Either they kill or they inhibit the pathogenic growth. So they also reduce the total microbial population, but on living tissue. So it is very important in the case of antiseptics, as they are used with the living tissues, that they should not destroy the host tissue. And it is important that the antiseptics, they should be generally less toxic as compared to disinfectant. The next term is the chemotherapy, which is the use of the chemical agent to kill or inhibit the growth of the microbes within the host tissue. That is again for the living tissues that we use the chemotherapy. So antimicrobial agent that kill the microbes uh, in the host tissue, we are going to use the chemicals for them, and the therapy that we call as the chemotherapy. Now we do have two terms, microbicidal and microbistatic. So there are antimicrobial agents that are going to kill the microbes, and such chemicals which kills the microbes, they are denoted by adding suffix side, CIDE, side. So we call it as a cidal agent that kills the pathogens except the spores, because spores are more resistant and they can survive in a better way. They have better chances of survival even in the adverse condition. So the antimicrobial agent that are going to kill the microbe, they are called microbicidal agents, while the antimicrobial agent that inhibit or prevent the growth of the microbes, they don't kill them, but they either inhibit or they prevent the growth rather than killing. So they are given, they end with the name static. So these agent, they are going to stop the growth of the microorganisms, right? So microbistatic and microbicidal, these are the two terms which are used in order to find out the chemicals which are going to either kill or which are going to prevent the growth of the microbes. Now there have been various factors, the various conditions are there that affect the effectiveness of antimicrobial agents. What different antimicrobial agents are we going to use? Either we are going to use disinfectant, we are going to use a sterilant, we are going to use a sanitizer, we are going to use an antiseptic. Whatever are we going to use, there are certain factors which decides its efficacy, and the first factor is the population size, right? So larger the population, it will always require a longer time to die as compared to the smaller one. So population size is directly proportional to the time taken to destroy the population. The second factor is the population composition. So the effectiveness of an agent, any chemical agent, it varies greatly as per the nature of the microbe being treated. Means what population we do have with us, because the microbes, they differ a lot in their susceptibility. Like the bacterial spores are very resistant to most of the antimicrobial agents than the vegetative forms. Vegetative forms are comparatively killed in an easier way as compared to their spore forms. So younger cells are usually readily destroyed than the mature ones. This is also one of the important factors in the population composition. Or we can say that some species like Mycobacterium tuberculosis, which is the causative agent of TB, are able to withstand the adverse condition in a better way as compared to the other microbes. So there are some of the microbes which are much more resistant to antimicrobial agent than the other bacteria. So in population composition, the nature of the microbe, that is their susceptibility, and the age of the culture, both are going to decide that what will be the efficacy of the antimicrobial agent. Third is the concentration or the intensity of an antimicrobial agent. Generally, the more concentrated chemical agents are supposed to destroy the microbes more readily. It is a general perception. But we can say there are cases that an agent is more effective even at low concentration, as we are familiar with the example of 70% ethanol, which is more bactericidal than 95% of ethanol, as the activity of ethanol is enhanced by the presence of water. So it is an exception where the concentration is not directly proportional to the antimicrobial concentration. Next factor is temperature. As we all know, that increase in temperature at which the chemical acts, it often enhances its activity. So at low concentration of disinfectant or the sterilizing agent, uh, we may use higher temperatures. Means at if we are going with the high temperature, we may have to use low chemical sterilant or low antimicrobial agent concentration. But if we are using it at a normal temperature, then there are chances that we may have to use the higher concentrations. The last factor, the last condition that is going to affect the efficacy is the local environment, as environmental factors around the microbes may either offer a kind of protection or they support their destruction. So we may have examples such as heat, which kills readily at an acidic pH. Means if we are going to heat the microbes, so at an acidic pH, heat will be a good factor to kill them. Acidic food and beverages such as fruits and tomatoes, they are easy to pasteurize than the alkaline food like milk. So here the pH is yet another local environment which is going to enhance the action of the antimicrobial agent. So these all are the different factors. We are aware of the different terminologies which are used for the antimicrobial agents. Now we do have different methods to control the microbes. Means what different ways are there by which we are going to control them, as the various physical, chemical and biological methods are employed to control the microbes. And as we already have discussed, the choice of method, it depends on the factors, that what type of microbes are we going to treat, what level of control is required, and the nature of the material being treated. The safety considerations are there, whether are we going to use it on inanimate object or we are going to use it on the living tissues. So all these are the factors that are going to decide that what kind of method will we use. So effective microbial control, it plays a vital role in preventing the disease and transmission. It also helps in maintaining hygiene, and it also protect the public health. So among the different methods, you have a mind map with you where you can see that mainly the methods have been categorized into two main branches: physical methods and chemical methods. So physical methods yet divided into three different zones: the heat treatment, the radiation treatment, and the filtration. That also depend on the type of material we want to sterilize. So we will select the method accordingly. Heat can be your dry heat or it can be moist heat. The dry heat is supposed to be less effective as compared to the moist heat. So heat and the other physical agents that are normally used to control the microbes, even in your day-to-day life, we use heat as the method of sterilization. And the most frequently employed physical agent as heat is being there. The second one is the radiation. We know it well that we do have ionizable radiations and non-ionizable radiations, which are used for the purpose of sterilization. You would have visited a dentist and would have seen there a UV lamp where they keep all their instruments. They keep their forceps, scalpels, so they are sterilized under radiations. You would have even thought of the syringe, the cannulas, how they got sterile. So they are not going to get sterilized by heat, or they are not going to get sterilized by using chemicals, but radiations are there, which is a way by which we can make sterile those packed items. The third one is the filtration. So when we are going to use the heat sensitive materials, we generally prefer for filtration methods. When we go to the second major method of sterilization, that is chemical method. So in chemicals, either we can use liquids or we can use gases. So in further discussion, we will discuss each of these methods in big details, and we'll try to explain the condition and the substances which we will take up in each of these methods. Now first is the physical method. As I told you, heat is a reliable and a rapid method which has been used extensively, and the heat was of two types: the dry heat and the moist heat. So dry heat is believed to kill the microorganism, and it causes the destructive oxidation of the essential cell constituents. Dry heat can be used at 100 degrees centigrade for 60 minutes, or it can be used for 115 degrees centigrade for 60 minutes to kill all the vegetative bacteria and fungal spores. But we all know bacterial spore being more resistant, they require even higher temperatures, around 160 degree for 1 hour or 180 degree for 20 minutes. So on the whole, this dry heat is supposed to be less efficient sterilization method as compared to the moist heat. So in the dry heat, you can see that we have mentioned here red heat, flaming, incineration, and hot air oven. The red heat is generally used for inoculating wires, loops, forceps, and spatula. We hold them in the vertical position in front of the Bunsen burner, which is shown here in the figure, and we we wait until they turn red hot. That is what is red heat sterilization. If I talk about flaming, so these cover slips, these glass slides, these mouths of the culture bottles, they are sterilized by passing the article through the Bunsen burner without allowing them to become red hot. Means we will just take it to near the flame and then we'll take it away. Means a continuous to and fro movement used to take place, so that the air which is going to enter into the flask, that remains hot and do not allow the easy entry of the microbe. The third one is the incineration. This is an efficient method for rapidly destroying the contaminated material, like the pathological material from the bio waste that can also be uh, treated by the incineration. And the last is the hot air oven. It is a method of choice of sterilization, especially for glasswares, uh, test tubes, and there are petri dishes which we sterilize through hot air oven. And in hot air oven, either we go for 160 degrees centigrade for 1 hour or 180 degrees centigrade for 20 minutes. So that depends that whether are we choosing the higher temperature range or lower temperature range, the time is adjusted accordingly. So this was all about the dry heat method. Now coming to the moist heat. So sterilization by moist heat means killing of the microorganism with steam, or we can say with hot water. And the steam, it causes the denaturation and the coagulation of the protein of the microbes which are being treated. So we do have the sterilization by moist heat at a temperature below 100 degrees centigrade. It could be at the temperature 100 degrees centigrade, or we can go for a temperature above 100 degrees centigrade. So we will take up the first case where we will use moist heat at a temperature below 100 degrees centigrade, which involves three main methods: pasteurization, water bath, and inspissation. So we all know that for the heat labile fluids, the disinfection is being done by heating at a temperature below 100 degrees centigrade in order to maintain their nutritional value, and we can preserve them for a longer period of time. So, such treatment is sufficient to kill the mesophilic vegetative bacteria, and these heat labile fluids like the milk, the butter, uh, the temperature which is employed is around 63 degrees centigrade for 30 minutes, or we can use 72 degrees centigrade for 20 seconds. So it is followed by a rapid cooling at 13 degrees centigrade or lower. So in this method, the non-spore forming bacteria, they are destroyed, while the spore forming bacteria cannot be destroyed by this method. Now moving on to the second one, that is the water bath. So in water bath, we are going to use it for serum, for body fluids, or we can use it for vaccine. So in a water bath, we are going to heat it at 100, means around below 100 degrees centigrade for 10 to 20 minutes. So it kills all the vegetative bacteria while spores are not killed. In case of the inspissation, I can say that it can be used to disinfect the media containing eggs or serum. The medium is placed in a slope of an inspissator and heated to around 80 to 85 degrees centigrade for 30 minutes, uh, and that too on 3 successive days. Means we are not going to give the treatment just for a single day, but for 3 successive days we are going to treat the media. So this is what we do at when we are going to use moist heat for sterilization at a temperature below 100 degrees centigrade. Now we have moist heat that we use at 100 degrees centigrade. We can go for boiling. So at 100 degrees centigrade, if we boil something for 10 to 30 minutes, it almost kills most of the vegetative bacteria. So the second method is the steaming, and steaming is done at the normal atmospheric pressure. We are going to use the free steam, and here in steaming, an Arnold steam sterilizer is generally used for the steaming process. The last is the tenderization. where we do go for 100 degree centigrade for 20 minutes, that too for 3 consecutive days. So this method is also known as intermittent sterilization or tyndallization. So when we first expose the microbes to 100 degree centigrade for 20 minutes on the very first day, all the vegetative bacteria and few spores are killed, while next day, being in a favorable medium, they will germinate and will be killed in the subsequent occasions. Therefore, the non-nutrient media cannot be sterilized by this method. So this is all about the moist heat at 100 degree centigrade. Now, when we are going to use moist heat above 100 degree centigrade, you may have a question in your mind that how can we go above 100 degree centigrade? So, steam above 100 degree centigrade, that we call as saturated steam, is a highly efficient sterilizing agent than your hot air, because it provides the greater lethal action, and it is quicker in heating of the exposed article. Even it can penetrate into the porous material such as the cotton wool stoppers, the paper, the cloth wrappers. So they it allow a better sterilization of the substance. So in this moist heat at a temperature above 100 degree centigrade, the sterilization by steam under pressure is performed, and for that we have an autoclave that we generally use. Here is a picture shown of an autoclave. So when the water boils, its pressure is equal to the pressure of the surrounding atmosphere, and when the atmospheric pressure is raised, then the is raised at a normal pressure, water boils at 100 degree centigrade. But when it is boiled in a closed vessel, an increased pressure, the temperature at which it boils and the that of the steam it forms, it rises above 100 degree centigrade. So when we are going to boil the water at 100 degree centigrade under a reduced pressure, the temperature rises above 100 degree centigrade. So efficient sterilization is achieved at 15 psi pressure, 121 degree centigrade temperature for 15 to 20 minutes. So this is how the sterilization is achieved in an autoclave. Now the second method is the radiation. We do have ionizing and we do have the non-ionizing radiation. The source of ionizing radiations are the X-rays, gamma rays and cosmic rays, uh, while the source of non-ionizing radiations are the infrared and ultraviolet radiations. The sterilization through ionizing radiation is also known as cold sterilization, because they have very high penetration power, and they are highly lethal to the cells including bacteria. And large commercial plants, they are used they use the gamma radiation for the pre-packed disposable items, as we discussed in the beginning, while the non-ionizing radiations, they are also used for the same purpose, but they are not that much efficient as compared to the ionizing radiations. The next we do have the filtration. For the heat sensitive liquids and gases, we go for filtration method, which is the simple removal of the microbes. To reduce the microbial population, we are not going to remove something here, we will just reduce the microbial population in a solution of heat sensitive material. It is used to sterilize the liquid and gases, including air. In case of filtration, there is no direct destroying of microbes, and we will just use the filters in order to remove them. So we do have mainly two types of filters, the depth filters and the membrane filters. The depth filters are made of the granular or fibrous material, and based on the type of the granular material, they have been named differently as mentioned in the slide. One of the examples of the depth filter which has very good application are the HEPA filters, high efficiency particulate filters, which are made from fiberglass, is an example of a depth filter. It can even remove the viruses that are zero point 0.1 micrometer or even smaller than that. The next we do have the membrane filter, which are supposed to be superior as compared to the depth filter, and these are the filters of porous membrane and a little over 0.1 millimeter thick, made up of either cellulose acetate, cellulose nitrate, polycarbonate or polyvinyl fluoride. They are available in a wide pore size, and they are used to sterilize the pharmaceutical, ophthalmic solutions, even antibiotics. The air filters are there, which are used to filter the air to remove the microbes, and the very good application during the COVID time, you would have seen the N95 disposable mask, which were used in order to prevent the entry of the virus into the body by using this mask. Then we have the cotton plugs on the culture vessels that are also a kind of air filter. Now, moving on to the next, that is the chemical method, which is used for disinfection and antisepsis. There have been a way that it should be effective but should have low toxicity for the living tissues. So first example of the liquid that is used as the chemical sterilizing agent are the phenolics. They were first widely used antiseptic and disinfectant. Cresols, xylenols, and orthophenol are the major disinfectant, and the phenolics act by denaturing the proteins and disrupting the cell membrane, and they remains active on the surface even after the long time of the application. One phenol is there that we call as tricolsan, which is used in hand sanitizers, as it can effectively block the fatty acid synthesis. So whatever hand sanitizers are you using do you use, it has tricolsan in it. So the second category of the chemical agent that are used for sterilization are the alcohol. They are disinfectant and antiseptics and are most widely used. They are bactericidal, fungicidal, but not sporicidal in their action. Popular alcohol germicides are your ethanol and isopropanol, which are generally being used, and they are used at a concentration of around 60 to 80%. And they work by denaturing proteins, possibly by dissolving the membrane lipids. The next category is of halogens, mainly two halogens, iodine and chlorine. They are important antimicrobial agents. Iodine is used as skin antiseptic, which kills by oxidizing the cell constituent and iodinating the cell proteins. Chlorine is a usual disinfectant for municipal water supplies, swimming pools, also employed in the dairy and the food industries. It can also be used as chlorine gas, sodium hypochlorite, hypochlorous acid, and it causes the oxidation of cellular material and destruction of the vegetative bacteria and fungi. But the problem with chlorine that it reacts with organic compounds to form the carcinogenic trihalomethanes. So the use is slightly restricted due to its carcinogenic properties. Now we have heavy metals, which are also used as the sterilizing agents. We have the mercury, we have silver, we have arsenic, we have zinc, which are used as germicides. Then we have the quaternary ammonium compounds. These are generally detergents with a broad spectrum antimicrobial activity, and they are also effective disinfectant. They have the ability to disrupt the microbial cell membrane, or they can easily denature the protein. Benzalkonium chloride or cetyl pyridinium chloride, they are stable and non-toxic quaternary ammonium compound. They kill most of the bacteria except the Mycobacterium tuberculosis and their spores. Next category, we do have the aldehydes, especially formaldehyde and glutaraldehyde. They are highly reactive molecule which combine with the nucleic acid and protein, inactivate them, and thereby working as a potent sterilizing agent. They are sporicidal and can be used as chemical sterilants. Formaldehyde usually dissolved in water or alcohol, while glutaraldehyde is effective disinfectant, which is less irritating than formaldehyde. So the use of glutaraldehyde is preferred over formaldehyde. At last, we do have the sterilizing gases, which is a kind of chemical that is used in the vaporized form. So heat sensitive items such as disposable petri plate, syringe, heart lung machine, the components, heart lung machine component, they are sterilized with ethylene oxide, which is the common gas which is used as the sterilizing agent. Ethylene oxide is microbicidal and sporicidal in its action. It is a strong alkylating agent that kill by reacting with the functional groups of the DNA and protein to block their replication and their enzymatic activity. However, the ethylene oxide is explosive, so usually supplied in 10 to 20% concentration mixed with carbon dioxide. The ethylene oxide concentration, humidity, and temperature, these three factors are going to influence the rate of sterilization. The next sterilizing gas is beta propiolactone, that we call as BPL. It is used to sterilize the blood products or vaccines. They destroy the microorganisms more readily than your ethylene oxide, and they do not penetrate into the material well and may be carcinogenic. So the use of ethylene oxide is preferred over beta propiolactone. Last is vaporized hydrogen oxide, which is used to decontaminate the biological safety cabinets and the operating rooms. The vaporized hydrogen oxide, also in short, we call it as VHP. With this, we have covered almost most of the topics, the most of the methods, how we are going to control the microorganisms. Hope this video is you will find it useful, and you will wait for the next video to come very soon. Thank you so much. Please like, share and subscribe.